Hi, good afternoon. As Lindsay said, I'm Claudia Mitchell, the CEO of Universal Cells, and today I'm going to present you how we're using genome editing to create the next generation of stem cell therapies. So at Universal Cells, we leverage our proprietary recombinant AAV genome editing approach to enable the commercialization of stem cell therapies. We believe that because our genome editing approach does not involve nucleases and not involve breaking the genome, we have the safest and most adequate genome editing technology for use in the clinic. We use recombinant AAV vectors to promote homologous recombination in a very precise way. We design our vectors to pair with very specific regions in the genome where we can introduce any site-specific changes that we want. So we use no foreign nucleases, and therefore there is no genotoxicity. We have no off-target cutting, and there are no on-target mistakes. Our delivery is very simple since our vectors are the delivery systems themselves. And finally, recombinant AAV vectors have already been clinically validated and tested in humans, making it an ideal clinic-ready manufacturing reagent. So our company is based on our proprietary recombinant AAV technology, and I'm subsequently going to explain how we use it to create universal donor cells that can be used as off-the-shelf therapeutics. So as you may well know, the commercial potential of stem cells is limited by host rejection due to antigenic differences in the different HLA genes. So currently there are two main strategies to overcome rejection. The first one is to use autologous or HLA-matched cells, but this approach is quite lengthy, expensive, and complex. So even though they're certainly feasible, they're not really commercially viable approaches. The second alternative that's currently being tested is the use of allogeneic cells. The problem with that approach is that it requires the use of immunosuppressive drugs that are known to be toxic and ineffective in the long term. So here I'm illustrating the differences between autologous stem cell therapies and the universal donor cell therapies. As I said before, the autologous stem cell therapies are quite lengthy and complex and very costly because each time you have to create a new clinical product. On the other hand, if you create universal donor cells, you only have to create one single master cell bank of pluripotent stem cells that can be thoroughly characterized, manufactured in scale. It avoids use of immunosuppressive drugs. It improves clinical trial design because you only have one cell line that you're going through trials. And in the end, it leads to more predictable and better patient outcomes. So how do we create universal donor cells? It is well known that the HLA system is the primary mediator of immune rejection of organs and cells. So there are two main classes of HLA molecules, class 1 molecules, which are expressed in all cells, and class 2 molecules that are expressed in professional antigen-presenting cells and also during inflammation. Class 1 molecules are formed by a heterodimer that is composed by the variable subunit, the variable chains, that are linked to the common subunit, which is called beta-2 microglobulin. For class 2, on the other hand, there are no common subunits, and you have a polymorphic heterodimer. So for class 1, there are 6 polymorphic class 1 molecules that need to be matched. So in the past, there have been approaches where the beta-2 microglobulin common subunit has been targeted to avoid all class 1 expression. But the problem with that approach is that NK cells recognize class 1 negative cells and kill them. So our approach was to knock in a decoy molecule that is composed by a single chain non-polymorphic HLA-E transgene fused with beta-2 that we knock in the beta-2 microglobulin locus to avoid the recognition by NK cells and still evade the immune system. So in order to fully control HLA expression and create universal donor cells, we use our recombinant AAV vectors to knock in, as I said, the non-polymorphic HLA-E transgene at the beta-2 microglobulin locus to replace all class 1 molecules by this single chain molecule. On the other hand, to eliminate class 2 expression, what we do is we knock out, using another AAV vector, the RIF-SANC transcription factor that is responsible and necessary for the expression of all class 2 molecules, so we don't have any class 2 molecules being expressed. As a safety feature, we can incorporate suicide genes during our editing process. So we usually use TK as a suicide gene, and we can also incorporate a second suicide gene if we want. The same way, we can also incorporate any additional genetic changes that we want during our genome editing process. I wouldn't have time to go through all the preclinical data that we have accumulated so far, but to summarize it, we have shown that we can create pluripotent universal donor cells by using the HLA engineering approach that I just described. We've shown that they do not elicit any immune response, neither in vitro nor in vivo in clinical and preclinical models. And we've shown that these cells hold great potential for clinical use since they do not differ in any way from the parental lines, apart from the HLA engineering, in that they retain full differentiation capacity. Also, we have established all the necessary quality systems to manufacture clinic-ready universal donor cells. So to talk a little bit about the company, we are a 3-year-old company founded by myself and by my scientific co-founder and CSO, David Russell. Our management team is also comprised by Gregory Block, our VP of Business Development, and we count with the advice from our 2 SAB members, Gordon Keller and Paul Fairchild, both leaders in the stem cell field. We also have 2 business advisors, Robert Coffin and Melissa Carpenter. We are now at 15 employees and counting, and we just recently moved to our new facilities where we have established our virus and cell manufacturing capabilities. In terms of our business model, we balance our pipeline between our internal and our partner programs. We are currently establishing manufacturing our clinic-ready universal donor iPS and ES cells, and that will be used both for our internal and our partner programs. We have so far established 2 strategic partnerships, the first one with Adaptimmune to create universal donor T cells, and we also partner with Helios to create universal donor RPE cells in liver and kidney organ buds. And we continue to actively engage new partners. At the same time, we're developing our own internal universal donor cell programs, and our goal is to take our universal donor cell internal program to the clinic in the next 4 or 5 years. So we have invested quite heavily in the last year to establish what we think will be the gold standard for genome editing of pluripotent stem cells. We have created an integrated quality system for cell manufacturing to meet stringent regulatory standards and all throughout our processes. It goes from the production of our plasmid that makes our viral vector for the gene editing to the final seed bank of universal donor cells. And we've done it all in-house to fully control the process, to accelerate cell line production, and to reduce costs. So as I said, our manufacturing is ready for the clinic. We have a high-quality production of vectors and cells. We perform very stringent evaluation of the starting pluripotent stem cell lines to make sure that they meet the regulatory criteria and also that they can be the basis for a BLA-worthy product. As I said, we have stringent quality systems for our genome editing process because we want to make sure that once the genome editing process is done, it's done right, and we have created a BLA-compatible cell bank that can be used for everyone for any indication. Regarding IP, we have full freedom to operate. We have more than 30 patents and patent applications filed that cover both our recombinant AAV technology, but also our HLA engineering approach, regardless of the genome editing technology used. So, for example, if another company wants to use CRISPR technology to do the same HLA engineering that I just described, it would still be covered by our IP. So to summarize, we believe we have the most advanced technology for editing the pluripotent stem cell genome with great accuracy, as we do it without nucleases. There is no off-target cutting, and we have a built-in delivery system. We have fully validated an IP-protected approach for creating universal cell donor cells, and we can use our recombinant AAV gene editing to add any other further genetic modifications in the cell. And finally, we have established our in-house universal donor cell manufacturing that can meet very stringent regulatory standards. Thank you very much.